These are the first set of your visual notes for cell communication. So the first thing I want to talk about is cells figure out what to do, what to make from signals from other cells. They don't just kind of spontaneously understand how to do things. They do receive and interpret signals. And those signals are critical to cellular function. So sometimes those signals just have to hop between adjacent cells. In this case, this is called local signaling. This is right from your textbook. An example of this is from embryonic development and from neurons. But basically, you have a cell that is producing, usually they're proteins, but sometimes they are lipids. And those proteins are exocytosed from the cell, and they diffuse through the bloodstream, they diffuse through the extracellular matrix, they are absorbed by the adjacent cells, and they produce some kind of response. Remember, the main movement for what we're going to talk about is diffusion, right? This is the main way that molecules move, and it's no different for cell-to-cell signaling, right? Remember, in the case of neurons, neurons don't actually touch each other when they send signals to adjacent cells, right? That signal is responsible for releasing neurotransmitters. Those are usually proteins that cause a response in the second kind of receiving cell. And those neurotransmitters diffuse across a space. It's called a synapse. We'll talk about that later on. But again, the motion here is just diffusion of molecules, and that's how this receiving cell knows what to do. It's because it received these signals from an adjacent cell, right? These signals can also be all across the body, not just with adjacent cells. So you might have the testes producing testosterone or the ovaries producing estrogen, right? And those hormones are released into the bloodstream, and then they travel everywhere in the body. Every cell in your body gets bathed in these hormones, but only the cells that have receptors for them actually respond. And this in testosterone mostly for men and estrogen mostly for women. And this is how your cells know kind of what to make, what to do, what kind of function they're going to have. It's because of these long distance hormones getting sent to them via the bloodstream. So there are three main steps in cell communication. The first is that you're going to have receiving of the signal, right? So here's your signal molecule, and it usually attaches to a protein receptor. Not always a protein, but for the most part, they are proteins. That protein or that receptor is going to have a shape change, and we'll see this more in a second. I'll do a little delta here for shape change. Shape change. And that's going to prompt a series of signals getting sent through the cytoplasm of the receiving cell, right? This is called signal transduction. That signal ultimately activates some kind of cellular response. So let's go ahead and turn to the big sheet. So let's start on this side of the big sheet really quick. If you zoom in here towards the bottom, I think this is the back of the big sheet, what you're going to see is exactly what we just talked about. You have a cell, you have some kind of receptor in the membrane of the cell. It receives a stimulus, some kind of signal, be it a protein or a lipid signal. Actually, sorry, I take that back. It'll be a protein. And that's the reception component. That's step one. Then you have transduction, and then you have some kind of response, right? So back to the other side of the big sheet, you have a model cell. This is going to be a liver cell. We'll talk about that in a second. But here's your model cell. Here's the cell membrane all the way around the periphery. And here are some groups of receptors that we're going to talk about. So this first receptor here on the left is called a G protein linked receptor. We'll talk about that in this video. Next video, we'll talk about tyrosine kinase receptors, and then we'll talk about ion channel receptors. And we'll also talk about lipid signals too. So let's start with our protein signals. So signals are also called ligands. This is vocabulary that you're going to have to get used to. Signals are also called ligands. So those two are interchangeable. Let me grab my marker. And typically, again, these signals are going to be sent via the bloodstream. But before we talk about the signals, actually, let's return back to these receptors. Notice that the receptor's embedded inside the membrane. So off to the side, I'm just going to do a little drawing. So here is the lipid bilayer, what I'm kind of hatching through right now. So here's the lipid bilayer, and here is your receptor protein. So let's stick with this one right here for the G protein linked receptor, right? The portion above and below the membrane, the extracellular matrix and the cytoplasmic side, those, remember, are hydrophilic portions of that receptor, of that protein, right? The middle portion is hydrophobic. So this was your receptor. And the portion of the protein, the receptor that's in the membrane itself, this hydrophobic portion, is called the transmembrane portion of the receptor. All right. Let's look at some properties of the protein signals that this receptor or these receptors are actually going to go ahead and receive. So the first, again, proteins are hydrophilic. Which means they can't move through the hydrophobic portion of the cell membrane, which means they stop at the membrane itself. They do not move through the membrane on their own. They actually stop at the membrane, which means you need, in order to receive a protein ligand, a protein signal, you need a membrane-bound receptor. Right? And for right now, we'll talk about this in a second. Let's just make one more note. These are fast and short-lived. So these are very fast, quick responses that, boom, immediately produce a response, and that's it. Let's contrast this with lipid signals, lipid ligands. So in contrast, lipids are hydrophobic. And because they're hydrophobic, they can pass through the hydrophobic fatty acid tails of the cell membrane, right? So they actually can go through the membrane. Go through membrane, which means their receptors are going to be in the cytoplasm. They won't be membrane bound. So receptor, and please add these notes to your big sheet, receptor and cytoplasm. And we'll see this in a second too. These responses tend to be slower responses and longer lasting responses. Longer lasting responses than your protein signals. And again, lipid signals also will travel through the bloodstream. All right, so let's look at a case study here. Let's look at a specific type of G protein linked receptor involved with adrenaline. Had the E there, right? So adrenaline is the same as epinephrine, right? Oh gosh, epinephrine. All right, so this is the same thing that's in an EpiPen, and it's involved in the fight-flight response, right? So adrenaline is a quick-acting molecule that gets immediate results. When you get a kind of rush of adrenaline, you have very, very distinct bodily characteristics that immediately happen. And some of these involve an increased breathing rate, it increased, this was an up arrow, sorry, heart rate. Your pupils dilate, a variety of other things need to happen, and that's because, so increased heart rate, that's because you need energy to respond. You need energy to flight, or you need energy to fight, right? Which means you need glucose. You need to release a lot, massive amounts of glucose very, very quickly. Right. So these signals, these G protein linked receptors and the signals they receive are going to produce fast responses, and I'll just say a lot, a lot of a very, very quick response. And since we're up here, we can go ahead and write this too. One signal is unique to one response, and we'll see this again too. But that response is very quick, and there's quite a lot of it. And since we're releasing glucose in this example, where is glucose stored, right? Well, it's stored as glycogen in liver or muscle cells. So this cell that we have on the big sheet it's a liver cell, or it could even be a muscle cell. Either type of cell will be just fine here because that's where glycogen is stored. While we're labeling things, and before we go through this, I do want you to label these molecules over here. And this is just to set the stage. So this circle is protein kinase A. Protein kinase A. And as is, it is inactive. I want you to write this. It is inactive. This triangle, and I'm going to abbreviate this, is protein kinase B. I'm going to abbreviate it as PKB, protein kinase B. Just like here, it is also inactive in its default state. This oval is glycogen phosphorylase. Glycogen phosphorylase. It's going to tell you exactly what it's going to do. And again, this is inactive. Parentheses there. This molecule here is glycogen. And I think that's good. That's good for that. So let's go back up here and we'll finish labeling this. A receptor right here is a G protein linked receptor. This is your G protein linked receptor. The one next to it with the G in it is actually the G protein. So this is the G protein, this is the G protein linked receptor. This kidney bean looking thing over here is adenylyl cyclase. So you have your G protein linked receptor, your G protein, and then you have this membrane bound catalyst enzyme, adenylyl cyclase. So these are all the players. We're in a liver and muscle cell. Let's go ahead and see how this process plays out. Let's draw our ligand. So this little triangle is our ligand. This is adrenaline. Here he is. That's the ligand. That's our signal molecule. And remember, it fits in the active site, or the reception site of this receptor protein. That is going to cause, or that binding is going to cause a conformational change in the cytoplasmic portion of this receptor. So here's there's the conformational change. I just drew a little shape change here. Go ahead and label that. So this is I think I might just type this one out. If it lets me, it's not going to let me. All right. This is a conformational, I'm going to abbreviate, conformational change, delta for change, in the cytoplasmic, it's not called the cytoplasmic portion, it's called the cytoplasmic domain. The cytoplasmic domain, or the cytoplasmic portion of the receptor after the ligand binds. After the ligand, in this case, the ligand is adrenaline, after it binds, right? So what does that do? This is going to hit, it physically comes into contact with this G protein, all right? It's going to activate that G protein. This G protein is going to get phosphorylated, and you're going to have to add this, by a molecule of GTP. We can add the phosphate group. It gets phosphorylated by a molecule of GTP, and GTP leaves as GDP. So very, very similar to ATP, but instead of adenine, a different nucleotide is being used. Guanine is being used, but it's still an energy molecule. So GTP is going to phosphorylate the G protein, it's going to activate the G protein, and then it's going to leave as GDP, right? Well, this G protein is now going to hit, because it's active now, it's going to hit adjacent adenylyl cyclases, right? Adenylyl cyclase is going to catalyze the reaction that follows it. ATP is going to get converted to cyclic AMP. So it's actually going to remove 2 phosphates. So let's actually scroll down and see what this does. So adenylyl cyclase catalyzes the reaction between ATP and cyclic AMP by removing 2 of the phosphates. Right? Cyclic, the C, just means that the molecule is actually in a ring. The molecule is like bent up on itself in a ring. So adenylyl cyclase rips off 2 phosphates from ATP and makes cyclic, which describes the shape, cyclic AMP. And cyclic AMP is very, very important, and we'll return to that in a second. Cyclic AMP is going to activate the inactive protein kinase A. Cyclic AMP activates the inactive protein kinase A. All right? So it's going to give it a phosphate. So here's the inactive pKa, and there's the here's the active form of it. Notice the phosphate is attached to it now. Thank you, cyclic AMP. You've done your job. That should be a check mark. And now pKa is active. Well, active pKa is going to phosphorylate inactive pKb. It's basically a game of pass the baton. You're going to pass the phosphate from one messenger to the next. So pKa passes the phosphate to inactive pKb, which activates it. So there it is. There's the phosphate group. So now pKb is active. And then pKb is going to add that phosphate to glycogen phosphorylase. So now glycogen phosphorylase is active. Remember, you're passing the baton. The baton is energy, right? This phosphate group is going to destabilize. It's going to energize these molecules. So you give it the phosphate group and you turn it on. You activate it. Well, active, remember this is active now, active glycogen phosphorylase is going to phosphorylate glycogen. It's right there in the name. So you're going to add phosphate groups to your glycogen molecules, and that will separate your individual glucoses. Right? So you get individual glucoses when all of this is done. And then these can very quickly go out into the bloodstream and then go off to cells that need it. This happens very quick. Fast. Right? So when you're looking at this, you might be saying, okay, well, why can't the cell go right from adrenaline this ligand straight to activating glycogen phosphorylase? Why do we have to go through all of this kind of stuff in the middle, right? And the answer is that if you do that, you can't amplify the signal. So this process going from the G protein linked receptor all the way down to free liberated glucose, everything in between, this is called an amplification cascade, or sometimes it's called a phosphorylation. A phosphorylation cascade, because you phosphorylate all these different molecules, and it's kind of like a waterfall cascade. And basically what that does is it takes one signal and it magnifies it, because there's a little bit we didn't talk about here yet. It's not that one G protein linked receptor only activates one G protein. Remember, these exist in a three-dimensional shape. So I'm actually going to switch colors here. One G protein, if you look at the cell membrane from above, one G protein is surrounded, one G protein receptor is surrounded by many G proteins. It's not just like there's one next to it. There's a whole bunch of G proteins next to it. So when one G protein receptor gets activated, has a conformational shape change, it's going to collide with and hit all of the adjacent G proteins. And all of those proteins are going to hit adenylyl cyclases, and so on and so on. So in terms of the actual numbers, one activated G protein linked receptor, just one, can activate 100 G proteins. That in turn can activate 100 adenylyl cyclases. Okay? One adenylyl cyclase can activate 10, can catalyze the reaction between ATP and cyclic AMP by 10 times. So by the time you get to cyclic AMP, you've got 1,000 cyclic AMP little messengers here. Just from one ligand, one adrenaline molecule gets you 1,000 cyclic AMPs. All right, so you're starting to see how this really produces a quick response. And remember, all of these molecules are diffusing around the cell. There's just a lot of them, so they collide very quickly, right? This signal, every step you take down the cascade, gets amplified by 10 times. By 10 times. So 1,000 cyclic AMPs times 10, by the time you get, I'm looking at my big sheet, by the time you get to your active pKa, you've got 10,000. So 1,000 times 10. By the time you make it to your next step down here, by the time you have active pKb, you do times 10 again, and you get 100,000 active pKb. Remember, this was pKa, this was pKb, and then each active pKb can activate 10 glycogen phosphorylases, so you multiply by 10 again. And when all this is said and done, 100,000 times 10 is 1,000,000. So because each molecule in this process can activate 10 molecules after it, by the time you're all the way down here with active glycogen phosphorylase, you have 1,000,000 active glycogen phosphorylase for one adrenaline molecule, right? And in terms of them activating or releasing the glucoses and glycogen, the total amount of glucose, this is the end glucose, is 100 million glucoses times 10 to the 8th glucoses can be released from a single adrenaline molecule, from a single adrenaline ligand. Quick, magnified in huge amounts, ready to use by cells that need to do cellular respiration immediately, right? So I would do your best. I mean, at this point you can pause, but go back, explain what's happening. Start with your ligand, go through your receptor, your G protein, your adenylyl cyclase, and then down through the phosphorylation amplification cascade, right? And before we end the video, I do want to talk about cyclic AMP and very kind of point it out and call it out, basically. Cyclic AMP is called a second messenger. And we're going to talk about second messenger molecules a lot in this unit. The first messenger, the one sending the message to the cell, is adrenaline. So this is your first messenger. This is your first messenger. So this first messenger is responsible for this initial kind of three-part response with your proteins in your membrane. But after that, you need a second messenger. So that response is immediately carried off with cyclic AMP. That's why cyclic AMP is considered to be a second messenger. Cyclic AMP is a second messenger in this phosphorylation cascade. Another common second messenger that you're going to see a lot of is calcium ion, right? That's another second messenger. Also, I'm going to go ahead and add this. Figure 11.16 in your textbook is a good reference for this. So figure 11.16. All right, and that's going to do it for this video. Actually, let me just add one more thing really quick, just because I forgot to do it. By the way, adrenaline is a product of your adrenal gland. So your kidneys are your renals, and the ad means on top. So there's a little gland up here. This is your adrenal gland, and it produces adrenaline, adrenaline. So I hope this video helped, and we'll proceed more with this next time.